Hi guys and welcome to my channel. Myself Dr. Akshay, and in today's session I'm going to be discussing about the topic lysosomes. If you guys want to learn more about the cell, its structure and its functions, then click subscribe and the bell icon. So let's do this together. Coming to the topic lysosomes. These lysosomes are vesicular structures, and these lysosomes are formed by the breaking up of Golgi bodies and eventually dispersing throughout the cytoplasm. This lysosomes provides an intracellular digestive system that performs the following functions. It digests the food particles which are engulfed by the cell. It digests the worn-out cellular organelles which are present throughout the cytoplasm. This lysosome measures about 250 to 750 nanometers in diameter. And while coming to the structure of this lysosome is composed of a lipid bilayer membrane. You can see this is the lipid bilayer membrane. And inside of this lipid bilayer membrane, there are so many granules which are present over here. And inside in these granules, there are 40 different hydrolytic enzymes. And these hydrolytic enzymes will split the organic compound into two or more parts. So the mechanism of hydrolytic enzyme is like this. So this is the hydrolytic enzyme, and you can see one protein molecule is approaching towards this hydrolytic enzyme. Soon after this protein molecule gets attached to this hydrolytic enzyme, with the presence of water molecule, this protein molecule will be getting splitted into two different amino acids. Here this hydrolytic enzyme will invite water molecules for the splitting process. In the similar way, carbohydrates are split into glucose molecules and lipids are hydrolyzed to fatty acids and glycerol. These hydrolytic enzymes are highly concentrated in these lysosomes. Usually this lipid bilayer membrane which is present over this lysosome will prevent the exit of these hydrolytic enzymes toward the cytoplasm, thus preventing the digestion of important cellular organelles which are present over this cytoplasm. And next, coming to PINOCYTIC and PHAGOCYTIC action by lysosomes. Thus, when the food substances start approaching towards the cell itself will form a vesicle-like structure. We can see here the cell itself is forming a vesicle-like structure. Soon, the cell will close that vesicle-like structure and a complete vesicle is formed. You can see this is a complete phagocytic or pinocytic vesicle that is formed. At the same time, these lysosomes will also approach this particular phagocytic or pinocytic vesicle and soon forming a digestive vesicle. And in this digestive vesicle, the carbohydrate will be getting converted to glucose, proteins will be getting converted to amino acids, and lipids will be getting converted to fatty acids. And these formed nutrients like glucose, amino acids and fatty acids will be getting expelled from this digestive vesicle towards the cytoplasm. Soon after the absorption of these vital nutrients by the cytoplasm of the cell, residual body will be formed. And this residual body will be excreted out of the cell by the process known as exocytosis. Thus, the pinocytic and phagocytic vesicle containing lysosomes are known as the digestive organs of the cell. Next, coming to regression of tissues. How regression of tissues occurs? This regression of tissues occurs due to the process known as autolysis. Here, lysosomes are responsible for this process of autolysis. So, some of those examples are, first one is the atrophy of skeletal muscles after prolonged state of inactivity. And the second one is regression of mammary glands after lactation process. And the third one is the regression of uterus after pregnancy. So these are some of the examples in which autolysis process occurs and thereby regression of tissues occurs. So how autolysis of damaged cells occurs by lysosomes? Here, damage to the cell can be caused by many different factors. They can be due to cold, trauma, heat, or other environmental factors. And due to these factors, it induces these lysosomes to rupture and release its hydrolytic enzymes, thereby digesting either a part of the cell or complete cell. If a part of the cell is getting digested, it can be repaired. But whereas if the whole cell is getting digested, it should be replaced by mitotic division to form new cells. These lysosomes also contain some bactericidal agents that helps in killing the bacteria. So some of those bactericidal agents are, the first one is lysozyme. This lysozyme will dissolve the bacterial cell wall. And the second one is lysoferritin. This lysoferritin binds with the iron and other substances and thereby preventing the further bacterial growth. And the third one is an acidic pH of 5. And this acidic pH will perform two different functions. The first function is it activates the hydrolytic enzyme. And the second function is it inactivates certain enzymes that promotes the bacterial growth. So that covers the topic lysosomes. I hope you guys learned about this particular topic. Thank you so much guys. Love you all guys. See you guys in the next session.